Hello everybody. Today my lecture topic is hepatitis viruses. Now, first as you can see, hepatitis infects the liver of the human body, and the chief character of which is the yellowing of the skin as also the yellowing of the eyes, as you can see in this slide. Now, before going into the details of what hepatitis is and what hepatitis viruses do to us, we will understand the root of the word hepatitis. It is actually derived from the Greek words hepat, meaning of the liver, and itis, meaning inflammation. So putting these together, hepatitis means inflammation of the liver. Now, of course, this is a medical condition characterized by inflammation of the liver together with the inflammatory cell accumulation within the tissues of this organ. Now, hepatitis can be self-limiting. What it means? It can heal on its own, or it may even progress to symptomatic cases, and that doesn't get healed up on its own. Two such severe symptoms will be: one, fibrosis, that means scarred tissues, and secondly, you have the cirrhosis part, which means development of nodules in the liver. Now, as you can see in this slide, you can see a fibrotic liver developed out of hepatitis viral infections. And if such cases of fibrotic liver and cirrhotic liver develops, then you know that at once liver transplantation is mandatory. Now, coming to the next slide, you can see the development of nodules in the liver. These are actually developed by hepatitis B and hepatitis C viral infections. So you can see the basic morphology of the liver underwent a major change. Now, if we compare the two histological sections, one from the liver of a healthy adult and another from the liver of hepatitis infected adult, you can see that here in the normal healthy adult, the cells are much organized and much closely packed together. But as you can see over here, the cells are much less organized, and also there are some spaces in between that indicates that some inflammation is taking place within the liver cells. If we now look at the causes of hepatitis, hepatitis may be subdivided into two principal types. One is acute hepatitis, which is sudden but very severe, and it lasts for less than 6 months. And another is chronic hepatitis that persists for even longer than 6 months. Now, if we come to the causes one by one, in case of acute hepatitis as well as chronic hepatitis, both viruses are the chief causes. And as you can find, apart from the viruses, alcohols, industrial solvents, some medications and autoimmune disorders are all responsible for hepatic diseases of the liver. Now, coming to the severities of hepatitis, sometimes hepatitis may be asymptomatic and remain so for decades also. Sometimes it may have limited symptoms, while sometimes it may develop some severe symptoms like jaundice, poor appetite, and a general feeling of malaise. Now, coming to the symptoms of hepatitis. Majority of the symptoms are matching with each other, like say for instance joint aches and malaise and overall discomfort. But what I want to highlight in acute hepatitis is that dark urine. This is a typical symptom of acute hepatitis, and this means that jaundice is underway. And if you go to the chronic hepatitis, you can see two very typical symptoms. One is the peripheral edema. Now, our main focus will be on viral hepatitis because it is the chief cause of hepatitis all across the globe. Now, this is a severe health problem and it affects several hundreds of millions of people all across the globe. Now, if we look at the causative agents, it is caused by a group of 5 totally unrelated hepatotrophic viruses. These viruses are of 5 principal types: hepatitis virus A, B, C, D and E. Now, as these viruses infects our liver cells gets damaged. How? Because our immune system actually puts up a protection against the viral infections. In the course of this immunological response, the liver gets inflamed, okay, that is swelled, and then the functions of the liver, which takes place normally, now gets impaired. So we will now look into the details of the hepatitis viruses. First, we will see a comparison of the 5 different types of hepatitis viruses A, B, C, D and E. But the first thing that I would like to highlight is their genome or the nucleic acid content. We will see that apart from hepatitis B, the rest of the 4 hepatitis viruses contain RNA in their genome. Only hepatitis B will contain DNA in their genomic part. Okay? And one more thing, in the next slide, we will see the mode of transmission that I will like to highlight. You can see that the viruses A and E actually transmit by enteral routes. And the viruses B, C and D, they get transmitted via parenteral routes. And then I would like to highlight on the diseases they cause. Hepatitis A causes infectious hepatitis. That means it can be contagious. Hepatitis B causes serum hepatitis. Hepatitis C causes non-A, non-B hepatitis. And similarly, hepatitis E causes enteric non-A, non-B hepatitis. Now, coming to vaccine, this is very important because you know that we want to achieve a protection against hepatitis, which is a deadly disease. So, there is a vaccination possible for hepatitis virus A and B. But alarmingly, there is no vaccination for hepatitis C. Now we will study the individual hepatitis viruses, which is our topic of discussion. Coming first to hepatitis A virus. As you can see, it causes infectious hepatitis. Now, this virus was first classified in the year 1983. And now today it is classified under the genus hepatoviruses. You can see how the virus looks like in the electron micrographic pictures. Now I can give you an example of a hepatitis A outbreak long back in the year 1988. So that outbreak was in China, in a place all of you know that is called Shanghai. In this place, you had an outbreak of hepatitis A virus in 1988. Now, coming to the characteristics of hepatitis A virus, it belongs to the picornavirus family. What is picorna? It actually comes from 2 words, pico and RNA. Pico means small and RNA, you know, that is the genome of this particular virus. Now, coming to the genomic part, the genome is a small, single-stranded and positive sense RNA. Now, the RNA contains 7500 nucleotides in it, and it is polyadenylated at the 3' end. If we go to the next slide, I can show you the poly A tail, which is located at the 3' end of this red colored thread that shows you the RNA genome. Now, going back to the previous slide, you can see that there is a polypeptide that is attached to the 5' end of this RNA. And what is the name of the polypeptide? VPG. I can show you this VPG protein attached to the 5' end of this RNA. This RNA genome actually encodes for a polypeptide. The molecular weight of it is 2,52,000 Daltons. This polypeptide is usually functionless in its native form, but it gets cleaved down into the essential protein parts of the virus, and then it acquires the functions. Now into two types it may be cleaved down. One are the structural proteins and another are the non-structural proteins for the virus. And the virus is actually surrounded by a particular type of capsid. The genome is surrounded, that is called the icosahedral capsid, and it has got a diameter of 28 nanometer. You can see that it is prevalent in the third world countries like India. Why? Because of conditions of malnutrition and poor senses of hygiene. So the route of transmission, as I have told you, is the enteric route, that is the fecal oral route. And how to contract this virus? Through the ingestion of contaminated food and water. Now coming to the incubation period of the virus, it is 28 days in our body before it actually begins to show the symptoms of the disease. Regarding the nature of the disease, it causes an acute hepatitis. That means a severe one but a sudden one. And chronic hepatitis is not caused by this virus. Now, what is very important is that the infected people excretes hepatitis A virus in their feces, which is 2 weeks before the development of jaundice and 1 week after the development of jaundice. Now, regarding the pathogenesis, what is most important is that it causes a necrosis of the parenchymal cells of the liver. And now the diagnosis part, you can always diagnose levels of AST. What is that? AST is aspartate transaminase, and what is ALT? That is alanine transaminase. You can also go to look for IgM and IgG. Now, treatment, only the people are advised to rest and to avoid alcohol. And coming to the recovery, the people usually recovers within 2 months, and 15% of the cases undergoes a relapse. That means the disease will occur once more. Now coming to prevention, which is so very much important, personal hygiene is absolutely mandatory, and you have to stop taking contaminated food and water. Regarding immunization, active immunization through vaccination can actually protect you for 10 years. Now we switch on to hepatitis B. These are also referred to as the Den particles, and they are the causative agent of serum hepatitis. You can see the structure of the hepatitis B virus under the microscope. Now it will cause acute hepatitis and chronic hepatitis, but acute forms are more common than the chronic forms. And it affects 1 to 2 million people across the globe, and majority wise it causes chronic hepatitis. Now coming to the family, it belongs to Hepatnavirus family. Actually it is HepaDNA. Hepatna means Hepa and DNA because it's a DNA virus. And you can see what is very important, the viral genome remains in association with a core antigen called the hepatitis B core antigen, HBCAG. And the entire genome and the HBCAG packed together remains within an outer envelope that comprises of the hepatitis B surface antigen, that is abbreviated as hepatitis B HBsAg. So if we look at the schematic diagram, you can see that the DNA part is actually not a completely double-stranded DNA. It is a partially double-stranded DNA associated with DNA polymerase enzyme. And you can see the red colored particles that are the hepatitis core antigens. And then at the outside part, you have the total envelope that comprises of the hepatitis B surface antigens. Now the genome is 3.2 kb in length, and it is comprised of 2 linear strands, that is straight strands of DNA, and the 5' ends of those strands are actually bonded to each other to give it a more or less circular structure. and one of the strands is incomplete. As I've shown you in this picture, one of the strands is incomplete. So this DNA genome remains in association with this DNA polymerase enzyme so that it can now finish that incomplete strand and make it fully double-stranded. Now coming to the host, humans and chimpanzees are the most notable host of this virus. Coming to the transmission, as I have mentioned, they get transmitted through parenteral routes like blood. Blood transfusion or abusive drug users, they actually contract the disease very well, or even sharing of shaving accessories like razor blades. And then you have tattoos, and you have a sexual mode of transmission also, and also via mother to child by breastfeeding. You can contract this particular virus. Now coming to the characteristics, its incubation time is 1 to 6 months within our body. So that is pretty long a time. And regarding the pathogenesis, it actually causes hepatocellular damage, that means damage to the cells of the liver, and it causes a viral clearance along with that. So we develop a chronic hepatitis, which is characterized by cirrhosis. And as also we have the development of a type of cancer, which is so shocking, and that is called the hepatocellular carcinoma, liver cancer. So regarding the diagnosis, it's very important because you can diagnose for whether any cancer cells are developing. You can look for the hepatitis B surface antigen, and as you look for that, you can find that antigen in your blood for about 1 to 6 months. So that phase is called the carrier phase, even after you have recovered from the disease. Okay. Now coming to the treatment, according to FDA, Food and Drug Administration of the US, you have these treatments like alpha interferons and entecavir. Now we will like to highlight on the hepatocellular carcinoma. Now what is that? Hepatocellular carcinoma is very dreadful a cancer because it is one of the 10 most severe cancer all throughout the globe. It affects our liver, and the persons who are actually HBsAg positive, that is positive for hepatitis B surface antigen, are at more risks of developing this cancer. So how this cancer develops? As this virus affects our liver cells will start to wither off due to the immune system reactions. So as the liver cells wither off, more and more liver cells starts to regenerate or replenish the lost cells, and they do it by cell division. Now as cell division proceeds, different checkpoints are there which controls the cell division, but here those checkpoints are not maintained. As a result, there is an uncontrolled cell division, and you have a lump of cells developed that is called the hepatocellular carcinoma, that is liver cancer. It occurs with hepatitis B virus mainly. Now coming to the prevention, yes, you have a prevention, but you have to take 3 doses of that particular injection, and that virus is a recombinant hepatitis virus, and that can give you a lifelong protection. Now coming to hepatitis C virus, it is called non-A, non-B hepatitis virus. You can see the structure of the virus under the electron microscope. It causes acute and chronic diseases, hepatitis, and affects about 3 to 4 million people per year across the globe. Now coming to the family, it belongs to the flavivirus family, but unlike the flaviviruses, they are not transmitted by arthropod vectors. Regarding the genome, they contains a single-stranded positive sense RNA genome. Now you can see the structure of the hepatitis C. You can see the single-stranded positive sense RNA genome. You can see the capsid surrounding the genome, and you can see the outermost envelope encircling the nucleocapsid part. Now This RNA genome, if I go back to this slide, actually lacks a 3' poly A tail. Now, this RNA genome also codes for a polyprotein. What is meant by a polyprotein? It can undergo a series of cleavage reactions and lead to the formation of functional proteins. Now, coming to the transmission, the route is parenteral. That means through blood or through placenta. But here I would like to mention that the HIV inflicted patients are more prone to getting infected by this HCV disease. Now the incubation time is 15 to 150 days, and regarding the symptoms, chronic hepatitis develops, which is characterized by cirrhosis. Coming to the treatment part, you have to take a combined treatment of interferons and ribavirin antiviral drug. Coming to the prevention, no, there is no vaccine for this virus. So the prevention is that you can avoid alcohol. But one thing you must keep in mind is that when people get infected with hepatitis C, they become more prone to get infected with hepatitis A and B. So you have to make them immunized to hepatitis A and B viruses also if they are not previously immunized. Coming to our fourth virus, that is hepatitis D virus. It is also called the hepatitis delta agent. Now, it is more similar to a plant viroid rather than a complete virus. Coming to the genomic part, it contains a RNA genome together with an antigen called the hepatitis D antigen, HDAG. And these together, RNA and HDAG, remain surrounded by an envelope containing HBsAg. Now, the genome is a single-stranded negative sense RNA. The RNA contains actually a protein message in that, which is referred to as the delta protein. You can see the RNA genome and the delta protein or the delta antigen that it encodes, and you can see the outer covering, which is made up of the hepatitis B surface antigen. Now, why it is called a defective virus? Because it cannot perpetuate within the hepatocytes without help from the hepatitis B virus. Hepatitis B virus helps it to replicate within the hepatocytes because it provides hepatitis D virus with the hepatitis B surface antigen part. Now the transmission is parenteral. Mostly the intravenous drug users get to contract this disease. That is through blood. It is transmitted. And it will lead to both acute and chronic hepatitis. The incubation period is about 1 to 2 months, 30 to 60 days. Coming to the last virus in our discussion, that is hepatitis E virus. As you can see that this virus is very common in our own country, that is India. And the first example of hepatitis E outbreak was reported as early as 1980. Now, it belongs to the family of Hepiviruses, which is very similar to calicivirus. Regarding the genomic part, it contains single-stranded positive sense RNA genome. It contains a 5' cap and a 3' poly A tail, and it encodes the structural proteins from its 3' end and the non-structural proteins from its 5' end. Now, as I have told you, this virus is very common in the Indian subcontinent, and it causes a enteric non-A, non-B hepatitis disease in the Indian people. The transmission mode is enteral, that is what? Fecal oral mode of transmission, and the incubation time is 15 to 60 days. Actually, this virus affects the young adults, but mortality rates are very high, about 15 to 20% in case of women, particularly when they are pregnant. Coming to the symptoms, what hepatitis E virus does, it imparts symptoms very similar to hepatitis A, and it takes a very major course in pregnant women, as I have told you, in the third trimester, and chronic infections may occur in immunocompromised patients. So, this is all about the discussion of hepatitis viruses in general. Hope you have benefited from this discussion and have gathered a knowledge how to cure yourselves and how to prevent yourselves from this particular disease. Thank you so much.